go through all the fungi groups now and go through each one of them and talk about them a little bit more. All right. So the chytrids are the first group, and they're kind of like the base group. They're really unlike a lot of the other groups, but their cell walls are made of chitin, and they do have exoenzymes like the other fungi. What's really different is they have zoospores, that is flagellated spores that move around, and they are in water, lakes, and sometimes in moist soils. And they're decomposers or parasites. They're you might have heard of them because they were in the news not that recently, but like last year sometime about they were parasitic to frogs and killing frogs in Central America. Anyway, their hyphae are a little bit different in that they both are haploid, can be haploid and diploid, but they're kind of hard to tell visually, and you can see, we'll look at them in lab and you will see probably what I mean by that. All right, so basically that's all I'm gonna say about them. The zygomycetes are a little easier to tell apart from other things visually. For one thing, they have a zygosporangia, and as we go through the life cycle, you're gonna see that has a very distinctive look to it. There's over 1,000 species in this group. They have really diverse life histories. There's some molds, so they'd be unicellular. There's some parasites. There's some commensalistic relationship with animals, and as well as decomposers. So let's go through the bread mold life cycle so you can see what their life cycle looks like. So a lot of their life cycle is going to be in this asexual stage, or this haploid part of the life cycle. That is, the spores will drop on the ground, grow into mycelia, and these mycelia are coanecytic. So what that means is they do not have a septum in between the groups. And then what can happen in the asexual form is that they will form these little sporangia that stick up. So they have, they kind of stick up, but they have a dark tart part to the top to them. So you might have seen that on bread, that is bread mold, has it looks like fuzz on the top, and that would be this asexual part. And that'll produce spores, and it'll grow in to more of that, and it'll just keep going really basically until the food source, that is the piece of bread, is gone. And then what it will do is do this sexual part of the life cycle. So what happens here is you have two mating types. So spores possibly from different mating groups that can be growing on the same piece of bread at the same time, but what will happen is those mating types will meet. So right here it's showing them meeting, and when they meet, they are going to undergo karyogamy. The plasmogamy, that is the actual cell membrane or cell wall of this guy is going to join with that guy, and the nuclei then can be mixed. Okay, so that's what's happening kind of right here. Okay, then what can happen is, so you have this group of nuclei from one mating group in the red, and the group nuclei from the other mating group in the blue, and what you'll get is karyogamy. And karyogamy is when those nuclei come together. All right, and at this point, what's going to happen is this zygosporangia will form. All right, so basically this then is diploid. All right, or at first it's not, and then after it actually does karyogamy, it is. Okay, so first of all, plasmogamy happens. These nuclei come together in those little structures there. Then that septum goes away, and so basically this is dikaryotic right here, or also known as heterokaryotic. And then, meanwhile, the zygosporangia is forming. All right, so if you see a zygosporangia, you're not sure whether it's dikaryotic or diploid. Okay, it could be either. But after After a time, of course, karyogamy, that is, the nuclei are going to fuse, form diploid nuclei, which will undergo meiosis and produce spores that are going to come out and either do this asexual life cycle again on another piece of bread, or they will go off in the wind and find some other food source. All right. So basically, that's that life cycle. All right. So there are some parasitic forms. They form microsporidia. These are kind of weird because they're parasites. They have no true mitochondria. They're using their host mitochondria, basically energy as an energy source, and they can be a risk to people with autoimmune deficiencies such as lupus or something like that, or AIDS. They can be used for biocontrol for insects, so that's another use for them. All right, so that's the Zygomycetes. Zygosporangia, very, very unusual or unique to that group. Okay. Glomeromycetes are what we call arbuscular mycorrhizae. Arbuscular is referring to the tree-like shape, tree-like shape of the hyphae that is within a plant cell. So these guys form symbiosis with plants, and there's over 160 species. These are septate. We kind of were talking about some of the Hostorium thing before. This is like a penetrative structure that is coming out of the edge of the hyphae. These guys are septate, by the way, and they will penetrate the plant cell, form their little arbuscular, which is a tree-like shape there, and they will live inside the plant cell. But what is symbiotic about it is the hyphae, of course, have a nice little safe place to live, and the nutrients that the plant is making, etc. And the hyphae themselves are picking up nutrients and basically like extra roots within the soil. So mostly this is going to be with plant cells that are part of roots where this is occurring, and these are called endomycorrhizae. All right. All right, so the next group is what we call Ascomycetes. Ascomycetes are called sac fungi. You're going to see why in a bit, because they form these little sac-shaped or cup-shaped things. There's 32,000 species of these, so a whole bunch of different kinds of these, and they produce bisexual spores in a structure called an asci. The fruiting body that holds the asci is called an ascocarp. In other words, that will hold many asci. There are some unicellular forms. They would be yeasts, but most of them are what we call cup fungi. Things that you might know are morels and truffles that are cup fungi. Some are plant pathogens, Dutch elm's disease, for example, chestnut blight, aspergillus, which is a lung infection, or you can get it as a lung infection. Most are saprobes, which means they are plant decomposers, and but about 40% of them will have a symbiotic relationship with green algae, forming something called a lichen. So we will talk about what a lichen looks like in just a second. Some are also mycorrhizae. All right, so let's look at their life cycle so we can talk about some of these structures. Okay, first of all, they would have possibly an asexual part, as most fungi do. And what will happen, of course, is they will, their mycelium, which are septate, will grow and produce these little things called conidia on the end of their structure. So the thing that produces the conidia is the conidia 4, and these have a look of like little teeny balls. I don't know if you can see that very well, but little teeny balls on the ends of each of these. So you'll be able to see those under the microscope in the lab. Those will come off and disperse, and they will just continue this asexual part of the life cycle. But then, of course, as in their sexual part, they're going to meet up with other mating partners. Okay, so they will have, or mating types, I mean. And what will happen is the mating types will come together, do plasmogamy, where it becomes dikaryotic or heterokaryotic. And in this case, what will happen is the ascus will start to form, all right? So these guys, if you can see closely here, it's got, in this case, the ascus only has 2 nuclei, one from each mating type, okay? And notice that when the mycelia came together, so mating type blue met up with mating type, they're not really showing it here, but it'll meet up with mating type red, and you will have this heterokaryotic cells with a septa in between each one of them, okay? So then what happens is as the original ascus forms, then you get karyogamy. So what happened then, you created a diploid nuclei, or the zygote, and then that will undergo meiosis. So when it undergoes meiosis, it creates 4 haploid cells, and then it will do mitosis and create 4 more, so there will be 8 haploid cells within an asci, okay? Now within the asci is in a dikaryotic structure. So this is the dikaryotic septum or hyphae here, and that hyphae will create this ascocarp, which is the cup, okay? And each one of those, that is these, right, these dikaryotic structures, is going to produce the asci, where karyogamy is occurring and creating 8 ascospores. So each one of those, that's one, that's one, that's one, that's one, etc. All right? So each one is producing the ascospores, and those are then haploid that will drop on the ground, and it can do the asexual part, or it can eventually go on and do the sexual part of the life cycle. All right? So that's their life cycle. This is a picture that's showing you the lichen situation. So what you've seen these, I'm sure, on the side of trees. There's different types of them. You'll see them in the lab. But basically, it's a fungi that is growing on the side of on the bark of trees. But it has a symbiotic relationship with green algae. And now this green algae we'll give it different colors depending on the color of the fungi and the mix with the green. Sometimes they're kind of grayish green, sometimes they're a little more yellowish looking, but anyway, they will have this symbiotic relationship. The green, of course, is doing photosynthesis. The fungi are saprobes, that is, they are using nutrients from the bark of the tree, and they are benefiting each other because they're both getting something that is either nutrients or a nice little place to live here. Now, what is happening in this guy, the way these guys, they can produce ascocarps, and that is, they would then produce the asci, and they could create more fungi, and then the algae would just infect them, so to speak. And the other way that they can reproduce is these Serratia. And Serratia are like little pieces of hyphae, right? This is little pieces of hyphae that are surrounding an algal cell. And those guys can break off, and they can fly through the wind, or basically, or they can drop them, drop down from where they are and land on another piece of bark and start another fungi. All right, so they will, once they start growing the fungi, the algae is already there, it reproduces, and you have a lichen that has been reproduced. All right, so lichens can do both. They can do the regular ascocarp the fungi produces, and then the algae will come in, and or it can reproduce by these Serratia. All right, the last group is called Basidiomycetes. These are club fungi, and they are usually on a stalk, so these are the typical mushrooms that you know. These are there's 30,000 different species. Shell fungi, which you might have also seen, they're oftentimes on the side of logs and so forth around here. Typical toadstools or mushrooms. There are some molds and mycorrhizae relationships. Some are parasitic to plants like rust and smuts. Some examples are wheat, black stem, or fusiform rust in pines. And but most of them are decomposers, like the shell fungi. And let's just quickly look at their life cycle. They also have a very unique structures as well. They have not really very commonly an asexual part to their life cycle. All right, so that's one big difference. So these are considered higher level. In the lab book, the Ascomycetes went last because then we talked about the lichens, which was kind of a separate group, so we just put that one last. But however, the Basidiomycetes is the considered higher level because it doesn't really have this asexual structure. Okay, so what is happening here is that the spores are going to drop on the ground and they're going to grow just like normal. All right, and these are also septate, and there will be two mating types, and there will be plasmogamy. That is the cytoplasm will fuse, and you will have these mating types that are coming together, making a dikaryotic structure. All right, so dikaryotic or heterokaryotic mycelia will grow into a reproductive structure called the basidiocarp. All right, so this is a massive hyphae with dikaryotic massa mycelia. I'm sorry, a massa hyphae called a mycelia, but it takes this form of this basidiocarp. All right, and it's dikaryotic. Then what's going to happen is underneath the gills, so you probably have seen underneath the mushroom, it has these little inundations, and you'll look at these more closely in the lab. But inside those gills, you're going to have a structure that's going to grow out that is also dikaryotic, but it's basically just hyphae extensions that grow into what they call basidia. All right. Then what will happen at the end of each of these basidia is that you'll get karyogamy, meaning that the nuclei will fuse and they become diploid. And then they will do meiosis, and they will form, each basidia will form 4 haploid nuclei, so 4 spores basically, which are basidiospores. All right, so this is kind of a cool up-close picture. It's not really quite showing the 4, there's one behind there, but 4 basidiospores that are then going to drop on the ground and form one of the mating groups. All right, so that's their life cycle. The asexual part can occur, but it's not as common, and it doesnant have any particular structures. So again, just to recap, our groups are chytrids, the zygomycota or zygomycetes, glomeromycetes, which are the arbuscular mycorrhizae, sac fungi or ascomycetes, and basidiomycetes, which are the club fungi. All right. Now the roles of the roles of fungi in nature are mainly as decomposers. There, as you've seen, there's some symbionts and some pathogens, some parasites even. Some pathogens to humans that we didn't talk about are like ringworm, athlete's foot, candida, which can overgrow your digestive system. And some practical uses are, of course, we can eat them, but also used in making cheese. They're a citric acid source. And, of course, yeasts are used in making wine, beer, other things like kimchi and that kind of thing. And then they are also medicinal.